and we have interleukin 2s. Helper cells help. What do they help do? The interleukins that they squeeze out make this helping stuff happen. So what they help to do is, one person said, it matures the B cell. Yeah, it matures the B cells. When B cells are fully mature, they become what? Plasma cells. And when plasma cells are fully mature, they give us what? Antibodies. So that's one thing. So that's a bunch of things that the interleukin 2 helps to do. Okay? T cells are still helping. One of the other things they help to do is stimulate the formation of B cell clones. The clones, we want to make B cell clones that are specific to those epitopes, right? So we want many B cells that are ready, engaged with their game jersey on and ready to go to work. So that's one of the things this helping feature causes to happen. You get a select clone of specific B cells that are ready to go to work. What else? Anybody else think of something else?
Do they turn one into a memory cell?
Yes, it stimulates the formation of a clone of memory B cells and
T cell.
yes, memory T cells. You've heard all of this before. We gotta start pulling this stuff together. That's why this particular the content in this exam is challenging, because everything is related to everything else. You can't just look at a slide and say, I memorized that slide. You have to start to pull things together. What happens and what does that mean, and then what happens next because of that? Everything, all of this, the innate and the specific immune system, all of this stuff is happening at the same time. And one thing relates to the next thing and to the next thing. So I want you to start, and we'll just keep doing this till you guys just start spitting this stuff out and say, of course I know what interleukin 2 does and what happens when a T helper cell, and this is what a T helper cell does. You're gonna know this. We're fresh out of the first exam, so don't stress, but I want you to start thinking about the way you're going to need to think to use this stuff. I'm not just teaching you this so that you can remember it. I'm teaching you this so you can use it. So you're actually going to know what's going on inside your patient's body to make immunity happen, to keep them healthy. And when stuff is not working, you can say, well, maybe it's this that's broken, or because the patient is doing this, or maybe it's this other thing that's broken. So you'll know how to approach a patient who's dealing with some sort of invasion, like flu or microbe. Okay, so this is a blow up of that little interaction up there. That's our APC, and that's our T helper cell. The APC has an MH2 class of molecule, and you know why it's not one. And it has taken its little piece of big antigen blue thing and broken it down and is presenting that particular epitope. This is the T cell that had a TCR on its surface that is specific for that antigen, so woohoo, it can go and connect to it. Now this CD4 thing, what in the world is that all about? Notice that when you look really closely, there's something more that's happening than just MHC antigen and T cell receptor connecting. Something else is connecting. And this CD4 little thing over here, receptor, is called a co-receptor. The co-receptor is on the T cell. And the co-receptor has to simultaneously recognize the MHC2 class that's connecting to this T helper cell. And you go, why? What? Why do we need something else? We know that this is the APC that connects to this T helper cell. So why doesn't the T helper cell just go ahead and start squeezing out interleukin and calling in the team to come is required to simultaneously recognize non-self antigens? Well, the reason for that is you know how when you're in a hospital or a patient's in the hospital and they need a blood transfusion and they order it up from the lab and it comes up to the patient's floor and the nurse comes in, but she can't do it by herself. She's got to get a partner in there. She's got to say, now look at her bracelet and look at the order and look at what the blood says and let's make sure everything is perfect before they hook it up to the patient. That's exactly what this is. Because if the one nurse came in and said, yeah, that's the antigen, and something was a little bit off, she had a headache, or she was worried about it was almost time for her to go pick up her kid from daycare, or they're going to sit them out on the step, and kid was going to run out in traffic, and she just worried about all kinds of things, and something was not quite right. This is the thing that is the second or the third nurse that's in the room. This must see MH2 before it allows the rest of this process to go on. If it doesn't recognize this connection, this MH2 here, it's going to say, I can't allow you to put interleukin 2 out there because I'm not sure it's not MHC1. It must know that this is something that is worthy of getting this process going. So you have to have a receptor, yes, but you have to have a co-receptor to say, okay, that's my fail-safe, I've double-checked, and I can call in the reinforcements now, and we can start killing stuff. So that's what basically the co-receptor is about. Remember when I said that there are some populations of humans who live very high risk, bad, putting themselves in harm's way lifestyles, the homosexual promiscuous males who are out there and they're in clubs every night, just a different person every night, and they've watched all their friends die of AIDS, but they don't get sick, they don't get AIDS. One of the reasons why is because they lack a particular co-receptor that's needed, and we'll talk about this more as we're talking about viruses and viral receptors. It will not allow the virus to come into the cell. So without certain co-receptors that are genetic, some people will not be able to succumb to infection. So these guys, these little co-receptors, look like they aren't all that interesting or all that powerful, but they are. So you have co-receptors that are beneath the surface that are required to say, okay, you can go ahead and call in reinforcements. Okay. Once all of these connections are properly made, we know this is something that is worthy of destruction. Activated T cells now can say, okay, all my interleukins, I can start calling out the rest of the army to do the work. Okay. Now, this is a slide that shows you all the things that we just talked about that T helper cells help to do. Once the B cell processes the antigen, gets it out on the surface, T helper cell comes in, says, yeah, I recognize that antigen, that epitope. The co-receptor is properly situated. It gets the okay to become activated. These are the things that happen. And you say, well, we said clonal expansion happens. We have an expansion of the B cells that are going to go ahead and get this work done. We're going to stimulate the maturation of the B cell line so that the plasma cells happen and they can start squirting out specific antibody to kill blue microbe. Memory cells of the B cell type are stimulated, and they go and they just hang out in the lymphoid tissue waiting for a second or subsequent exposure to blue microbes. And now you have some new words in here that we haven't talked about before. The interleukin-2, in order for these things to happen, what they do is they stimulate growth and differentiation factors inside the B cell. So all these mitotic divisions start to go, and that's how the clones start to clones start to expand. Okay. And the maturation, more and more divisions give rise to the plasma cells. Just sort of gives you another peek beneath the surface. Okay. Now, these are all things you know already. You wouldn't have known this yesterday, but you know this now. Extracellular antigens floating around outside. B cells, receptors on B cells are what? Receptors on the surface of B cells are what?
Antibodies.
Antibodies. What class of antibodies? IgG and
IgM.
what kind of M? Monomeric. The real IgM that we make when we're first exposed to an antigen has how many parts? 5. IgM is a pentamer, except when it's acting as a receptor on the surface of B cells. Very good. Okay, so this B cell recognized the square antigen as the one it was born to respond to. It brings it inside, it crunches it up into little pieces and epitopes, and they hook up to MHC class 2 and endoplasmic reticulum and all that stuff that you already know, Golgi, and it presents it to the outside of the cell. Specific T cell can recognize the epitope on the surface, hooks up to it. You don't see the co-receptor, but you know it's there. And when everything is okay, the T cell helps by squirting out the cytokines and all the things you know that happens. This only shows plasma cells, but you know there are more things that those cytokines do. You know all of this already too. Just different ways of looking at it, and I particularly like different ways of seeing it, different ways of describing the things that I think I already know. And when you read these kinds of things in the book, it'll add little things that we may not have talked about and say, oh yeah, okay, I get it. Okay? We know that the T cell up there, it's going to be our antigen presenting cell. And we know that the antibody types are D and monomeric M. Microbe up there at yellow circle 1 is recognized, brought in by endocytosis. Beat them up, break them up in yellow circle 2, connected to MHC 2, presented to the surface, specific T helper cell with its co-receptor that you see in the little box now. You know what all of that means. When the T helper cell has made the connection and everything is okay, it starts to secrete cytokines, and one of those must say that somewhere. And we have activated B cells. We get a clone of activated B cells. We get a clone of memory cells that go hang out in the reticuloendothelial system for another day, another week, another month, another year, decade down the line when we might come into contact with microbe up there again in number 1. All the increased mitotic divisions and all that stuff, we get the maturation of plasma cells, and they make IgM antibodies. It's IgM here because these are the first ones, the first isotype of antibodies that the body makes after it has first been exposed to a new antigen. antibodies that we make, then go and find more of those microbes up there, attach to them, destroy them. And now your patient can start to be better. And you see that 1, 2, 3, 4, 5. This shows you a pentamer of IgM, and we'll talk about why pentamers are really handy and very cool to be the first responders. Okay. We talked about antibody or immunoglobulin anatomy already. So they look like Y's, and I stand up here and look silly like this all the time. And the hands out here, what happens at the hands? What kind of receptor? Huh? The antigens attach. So my hands have which heavier lights are out here? Both. Because my big heavy chain is one of the heavy chains goes the whole way, all the way from constant to what constant, and so what's out here? Variable. And the variable areas are my fingers. And my variable fingers out here of my antibodies, if it's not specific for you, it's not going to stick on you. Not specific for you, it's not going to stick on to you. If it's specific for this guy, I got him and I'm taking him out. All right? And if I'm an IgM, then my other IgM friends, I got 5 ways to hook up to this guy, and we're really going to take him out. Okay? All right. FAB stands for which part of me? Which part of the antibody? Huh? Go ahead. FAB. FAB. And then what's the other one called? FC. Now, AB, think of AB and you'll never get this wrong. It's antigen binding. If it's antigen binding, it must be my fingers, right? This is specific end. My feet are the FC, constant. All of the bottom part of the antibody is constant, nothing variable there. All the variability and the specificity are in my fingers. The FC is what stands on various cells, like the B cells. The FC portion is what stands up on the B cells. Okay. Here I am again. It's the FC portion. All of the FC portion is constant. All of the finger portions at the ends, the antigen binding, the FAB ends, are variable. Now let me tell you something about these constant regions. All right, we haven't talked about the different isotypes of antibodies yet, but we will. So we know about IgM, because we just talked about them. We know about IgD. There's a few more other kinds. What other kinds are there? E? Huh? G? A. Okay. Now, what's important for you to know is that all of my, I'm the host now, all of my IgD antibodies in my entire body share the same constant binding, okay? And it will bind only to specific antigen. It fits. This one, the green fits. The blue won't fit. The purple won't fit. There's probably some antibodies somewhere in our circulation that will, but this one has no effect against any other antigen. This is an interesting one. Oh, no, this one. Porphyrin. When we learn about vaccines and what we learned over time were the farmers who cared for and were in close contact with these cows, when smallpox came around, they didn't get too sick because they had been exposed to the cow form of the infection. They didn't get cowpox because they weren't cows, but their immune system still made antibody against these organisms that the immune system had been exposed to. The antibodies that they made were specific for cowpox infection, but it was similar enough to the antigen of smallpox that those farmers had some protection against smallpox. So they did better than anybody else, the other people who farmed corn or something like that. Because although it wasn't a perfect fit, it was a pretty good fit. There was enough of a fit to give them some protection. So that's how poor fit might come into play.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
You see that even though we're talking about different antibodies that are being produced, it's the gamma has the constant chain here, and even though these are 2 different antibodies against 2 different antigens, they're still IgGs, so the constant regions are the same. That's basically what I want you to see. I'm not going to ask you to describe how we make an antibody change. We have to, have a B, pick one from the B, pick one from the D, and the J. I wanted you to see what happens inside the slot machine, but I don't need you to be able to make a slot machine, how to fix a slot machine when it's broken. You just want to read what comes up on the screen, okay? So I do want you to know that this is why all the gamma chain, all the IgGs have the same constant region, and that there is a method to the madness, but you do not spend time on this madness, okay? All right. Okay, now we've talked about this. This is stuff you already know. We gotta beat it into your brain to make it sticky. Okay, the FC portion is which part of the antibody? Huh? The skin. the constituent that doesn't have any B parts, it's all C, okay? And it's the part that stands up on macrophages, on neutrophils, on eosinophils, on mast cells, on many cells have antibodies associated with it. And the part of the antibody that attaches to those cells is the Fc piece. Certain antibodies have regions on the Fc portion for fixing complement. If you think way back, 2 classes ago, from when we learned about complement, we said we need how many molecules of antibody to make the classical chain go?
Wait.
We need antibodies in the classical pathway, right?
Oh, okay.
Classical pathway is the one that's antigen antibody, right?
Yeah.
Okay, and will one antibody molecule do it? Get it going? You need how many? You need at least 2, okay? Now, if the floor is the bug that we're trying to kill, and complement's going to help us kill it by the classical pathway, am I going to stand up on the microbe like this, or am I going to do a handstand on him? Huh? I'm going to do a handstand on him. So I need, you got to do a handstand on him, and I have to do a handstand on him. We both do handstands like this. And the complement attaches to our feet. Remember? Now it's going to be a great picture. So this is the bug. This is me doing a handstand. It's you doing a handstand. And what happens here? What's this?
Complement.
Complement what? See what?
1.
C1, yeah. Okay. So what this is showing you, and why I'm spending so much silly time on this, is to show you that where is it? The second thing, certain antibodies have regions on the Fc portion for fixing complement. So this is the Fc portion up here, because the FAB portions are doing a handstand on the bug, right? So it's the Fc portion of the antibodies that stimulates the classical complement pathway. Okay? Binding Fc, release the cytokines. That's for the mast cells and stuff, and we'll talk about that soon too. Okay? We at least mentioned in passing that there are 5 different isotypes of antibodies, and these are they. IgG is a monomer. Plasma cells make them. They are produced in a primary response, which means it's this response to the body after initial contact, first time contact with an antigen. It's also produced by the memory cells just hanging out in the reticuloendothelial system. They're going to squirt out IgG, and IgG is the most prevalent, present in the highest concentration in the circulation. Okay? IgA is a monomer, and it circulates in the blood, and it's a monomer when it circulates in the blood, but it's a dimer when it's in our mucous membrane. Okay? So it comes in 2 pieces when it's in our mucus or serous secretions. Okay? And its claim to fame is being secretory, or in our secretions. IgM, we know, is a pentamer made up of 5 monomers, and it's the first class of antibody that's produced when we first get exposed to a new antigen. IgM comes first, and IgG comes later. IgD is a monomer. And its biggest claim to fame, if you look, you'll find other things that it does, but for our purposes, it is the receptor on the surface of B cells. IgE, we know, is present in higher concentrations when we're having a hypersensitivity or allergic reaction to something, and when we have a parasitic or helminthic worm infection. Okay. Now these guys, M and A, are the ones that are a little bit more complicated in terms of their anatomy. They have the same basic anatomy that we've already talked about. Long chains, light chains, constant regions, variable regions. All of that stuff is the same. Okay? The difference, as we already spoke about, is that IgM, when it's produced in response to an antigen in a host, a new invasion in the host, has 5 pieces. And they are connected at their feet. They're connected at the FC portions by a J chain or a joining chain. It makes sense that they're connected at their feet because they want their hands to be available and free to make contact with this thing that they're trying to get rid of. Okay? So if you just think about it logically, I can't trick you into saying that they're connected by their hands. They're holding hands. No, they're not. They've got their feet all hooked together by the J chain. So they need their hands to be free. IgA, or secretory IgA, comes in 2s. It's a dimer. Same Y anatomy. Also connected at their feet, at the FC portion, because they need their hands to be free too, by a J chain or joining chain. And the J chain, the J piece in secretory IgA allows the antibodies, the Ys, to be stable in secretion. Secretions have all kinds of stuff in them that protect us innately from things that it doesn't want to be there. The J chain helps the IgA not to be destroyed when it's in mucus and serous secretion. Okay? And there is a says stabilized to resist enzymatic digestion in body secretions. The body secretions, and these are some of the secretions where they are: milk, tears, saliva, mucus, but when it's in blood, you know that it's not a monomer. It's not a dimer. It's a monomer. So we got IgMs connected by their feet, hooked together by a joining chain. Secretory IgA is the dimer joined together at their feet by a J chain. But it also has a component in it that allows it to be stable in secretion. We're still okay. Okay, we know all of these except the only thing we haven't talked about here is the last bullet point. Okay, the FC region. We know that the character of the FC region is determined by what class of antibody we're talking about. If we're, the FC region of an IgA is going to be different from the FC region of an IgG. And we know all of the beneath the surface reasons why that is. We know that the FC piece is what activates the complement pathway. Notice that we're getting some more information here. Don't let it slip past you. Because because if I ask you what class of antibodies are capable of activating complement, I want you to tell me both IgG and IgM have the capacity to activate complement. The other ones do not. If the other ones do, it's in very, very small, has very, very small activity, and we're not gonna worry about those. And so IgG and IgM can activate complement. Fc portion of IgG can bind to phagocytes, like our neutrophils and our macrophages. And IgE binds to mast cells, basophils, and eosinophils. These are our granulocytes that squeeze out bad stuff. Sometimes it's good, but when it squeezes out too much of it, that's when we're talking about hypersensitivity, and you have allergic type reaction. So the IgE binds by its Fc portion to all those granulocytes, and phagocytes have IgG on their surface. Natural killer cells have IgG on their surface. Okay, let's take our break.